everybody. Thank you for joining us for this week's Lunch with the Scientist. Today, we are joined by Dr. Tonya Shearer. She is a PhD in biology and specializes in coral reef health using genetic and genomic technology. As a marine biologist, her research led her to study reefs all around the world. And most recently, she has joined the Guy Harvey Ocean Foundation as the Director of Strategic Partnerships to develop marine education programs with academic, government, and commercial partners to share her mission of ocean conservation. So with that, I'm going to go ahead and bring in Tonya. Hi, Tonya. How are you?
Hi, I'm great. How are you?
I'm doing fantastic. Thank you so much for joining us today.
Well, thank you for having me. I'm excited.
So we always like to start our Lunch with a Scientist with our scientists kind of sharing how they got into their career, what motivated them, what inspired them. So would you mind telling us a little bit about how you got started?
Sure. People always ask me, a lot of people say, I always wanted to be a marine biologist, or my kids want to be a marine biologist. And so my story leads back not to when I was a little kid. It was actually in college when I realized that I was going to be a marine biologist. I was always good in science. And so I took lots of science classes. And one of the classes that I took was called Natural History of the Caribbean. And as part of that course, we took a field trip. So I went to a school in New York, and I've been to the ocean on vacation or, for short visits. But this trip was going to take us to Jamaica. We were going to a research field station in Jamaica as part of this course. So before we went, several of us got scuba certified. And when I went diving in Jamaica, I decided at that moment that I needed to be paid to go scuba diving for the rest of my life. So that's how it turned out. That's was the first thought of being a marine biologist during that dive trip. So I guess I'll just jump into what I do now. So I have lots of hats that I wear, as most adults do in their jobs. I am a marine biologist by training. I am a molecular ecologist by training, and I'll go into what that is in a moment. I am an educator, and I develop educational programs, and I'm also a business owner. So a marine biologist, people know that I study organisms that live in the ocean, right? Molecular ecologist, people don't really know what that means. Ecology is sort of the studying the interactions between organisms and their environment and other organisms. And I do that on a molecular level. Molecularly, I'm looking at the DNA and the RNA of the organisms to see how one is impacting the other at that scale. And I'll show you more pictures of that in a little bit. So DNA and RNA are the big targets when I'm working with molecular ecology. Let me show you a little bit more. So I started, that trip that I took was from this college called SUNY Fredonia. It's in Western New York. It's just a small little college. I went there for 4 years and got my bachelor's degree in biology. Once I decided I needed to be a marine biologist and get paid to go scuba diving for the rest of my life, I went to Louisiana State University. Go Tigers, any Tigers fans? I went there for my master's. I was there for 2.5 years, and that's where I started learning the genetic techniques. And I learned a lot more about marine science there. I went diving. There's some cool diving places I went in the Gulf of Mexico and in Florida. I did some studies in Panama briefly as part of that project. After I got my master's, I went for my PhD, and I went back up to New York, to Western New York. Even though there's not a lot of ocean, there's no ocean, there's no corals up there, there are several marine biologists that work at University of Buffalo. And so I got my PhD there. We spend all of our summers in Florida or in the field. I learned more about the genetic technologies that are being used to study populations and organisms. I learned a lot about diving and reef studies. the organisms that live there. And so I got my PhD there. That took another 5 years. And then once I got my PhD, the next step is to do a postdoc. It's sort of like a job you get after you finish your PhD. And I got that at Georgia Tech, Georgia Institute of Technology here in Atlanta. I currently live in Atlanta. And I started as a postdoc there. That's what it's called. And I started working in Fiji. You'll see some of the work that I did in some slides later on. And I worked there for almost 10 years. I started as a postdoc, and then they hired me on as a research scientist. So I studied there for a long time. After about 10 years, I decided I wanted to do something else, and I wanted to get into education. And so I opened a science, like an informal science center called Discover Science Center. I knew that the schools needed some help teaching science, and we have a lot of homeschoolers around here. So I was teaching homeschool classes. And I had also, as part of my work at Georgia Tech, worked in the schools, bringing aquariums into the school and teaching the teachers how to use that as part of their STEM curriculum. So I knew I could create a job doing these types of things. And so I did. And so we've been open now for almost 9 years. And I have 2 locations. So I run those businesses. And I have a great team that teaches tons and tons of kids every year. We do summer camps and after school programs and homeschool classes and Girl Scout workshops. Most recently, within like a few months ago, I was recruited by the Guy Harvey Ocean Foundation. I am now the director of strategic partnerships there. And my job is to kind of do what I did with Discover Science Center. I develop education programs and curriculum around ocean conservation. And we use that curriculum and that framework in different places in Florida. And we're extending that into to other states and eventually other countries. So I kind of do what I do at Discover Science Center, but on a larger scale for the Guy Harvey Ocean Foundation. So when I am in the field, this is my second office. I'm very lucky in that I get to spend a lot of time in the water, at least I did when I was doing a lot of research. But what does that look like? So I figured I'd talk a little bit about what we do when we are in the field. My job in the past is more like field work and lab work. Now I have a lot of office work too. But field work is where I like to spend a lot of my time. And scientific diving is the way we get to work. So just like most people get in their car and drive to work, scuba diving is the way we get to our job site. And we use this term scientific diving as not just regular scuba diving, but we have a job to do once we're underwater. So it's very task oriented. And I put some pictures up here. So there's one here with this big rectangle. That's a tool that we use to help us isolate an area. This is actually on a shipwreck. It's a sunken, it's not really a shipwreck. It's an artificial reef, but it is a ship that they sunk. And one of the projects that I am involved with or have been involved with is looking at an invasive coral. And so this is how we collect our data. We count how many of those corals are in this rectangle, and we do that over and over and over again across the surface of the wreck. Because it's so big, we can't count every colony. We do a lot of sample collections and collect a lot of data. Lots of data, lots of samples in these bags. Do a lot of measuring here. There's a coral here. This is one that we've tagged and we return to and take pictures to see if it has gotten bigger over the year, gotten smaller over the year, if there's any disease that has shown up. So we map everything and then we come back and look for these little tags, and we use these measuring tapes to help us relocate. Down here are some recruitment plates. We put these little tiles out and we let little baby corals grow on them. And then we can see how many little babies are in the area and what species they are, and just some more measurement here. So these are just some of the tasks that we do as scientists underwater. So when we go out on in the field, we don't always go on little boats. Sometimes we go out in big ships. This is one of the ships that we spent a lot of time on, doing a few projects in the Keys and in the Gulf of Mexico. This used to be the ship that the Environmental Protection Agency owned, called the Bold. It's a fantastic ship. We would stay on there for a week to 2 weeks, and they would take us along the Keys and go wherever we want. We live on the boat. The crew is amazing. There's a lot of people that work on the boat that make sure the scientists can get the job done. We don't drive the boat, right? The captain drives the boat, and he has this crew of 17, and they make sure the scientists are able to get all their work done once they're offshore. We can't dive directly off of this boat. There's a big drop. This is where we would come off the back of the boat. So, and it's really hard to get back on the boat with scuba gear. So we go out in these little dinghies. They lower these down and we all get in. This is one of my dive teams. So as a scientist, especially underwater, as a scuba diver, we can't stay underwater all day long. We don't have enough time to collect all the data we need individually. So we have a team of divers. It's sort of like citizen science. These guys are not scientists, but I've trained them as to how I want the data collected and what samples I need to be collected. So when I'm out on a ship, I'm the chief scientist or the principal investigator. And my job is to train everyone on how to collect the data that I need for my research. So these guys go out. I send them out to go collect the data, go collect the samples that we need. And I go out as well with another team. This is my group. I'm here in the all black, all the way on the right. My divers are making sure we have all of our scientific gear. So when we go in, we don't have to come back up. We can be as efficient as possible when we're underwater. It's really expensive to do research out in the ocean, and we don't have a lot of time. So we're trying to be as efficient as possible. So I am going to share a little video. Let me give you a little bit of reference here. So this is in the Gulf of Mexico, kind of away from the shore, but this is on an oil platform. We're in about 400 feet of water, maybe 300, 400 feet of water. So there's no bottom that we can see. And we're only about 30 or 40 feet down. So we're not very deep, but below us is much deeper. So you'll notice everything is attached to us, because if we drop something, it's going all the way down to the bottom. We're not going to be able to recover it. So everything's kind of attached. People tend to think that oil platforms are like really dirty and nothing's living there. And during the video, you'll see fish. This whole area is covered in live organisms. These are all sponges and corals, and there's oysters. There's lots of fish around. So you'll notice that there's a lot of life in that area, even though we are on a working oil platform collecting these samples. This video is taken during a research dive where we are looking at an invasive coral. It looks yellowish here, but when you take it to the surface, it's orange. It's called orange cup coral. And I look at the genetics of this invasive species. This is not something that most people do when they're scuba diving. So if you were ever scuba diving, please do not take a hammer and chisel and break corals. It is illegal. I have a permit to do it, so I am allowed to do it under certain conditions. So this isn't something that everyone does once they go diving. So I'm going to show this to you. So here I identified the coral that I wanted to. So these are those yellowish and orange things are the corals, and I'm measuring it with that little measuring tape. I have 4 divers here. I'm the one that's doing the measuring. I am showing the diver that you can't see. She's writing down the size, and then the diver that you do see, he's going to help me collect. He has little Ziploc bags that when we take the small sample from that colony, he's going to put it in the bag, and then we'll take it back up on the ship and preserve it so I can do genetic techniques on it. So he's showing her the number on the bag. You can see all those fish in the background. So she's writing that down. And then the fourth diver is the person videotaping us. So we have a dive team of 4. The most common question I get after I show that video is, how do you write underwater? And this little white thing that she has here, that is a dive slate that is sort of plastic. It's PVC, and you can write with a regular pencil. We also have underwater paper that it's made of plastic. So it feels like paper. You can write on it like paper. You can print on it like paper, but it doesn't rip and it doesn't fall apart when it's wet. But that's how we're able to do it. I get that question a lot. So these are all the corals. And then these are sponges in between there. So everything's alive in this shot that's on this beam of that oil platform. I'm going to show one more video. This is on an upright, one of the legs of the platform. Same thing we're doing. We're selecting the coral. You'll notice that my camera, when you see me, is attached to my arm. My measuring tape is inside my sleeve so it doesn't drop to the bottom. But we have to be really good divers here, because we're not standing on anything. We're not kneeling on anything. We're neutrally buoyant. So we're able to control our depth just with our breath and our equipment. So here I am just taking a piece off. We always go with 2 sets of gear in case one of us does drop our hammer or our measuring tape. So someone else is carrying an extra hammer so we don't have to end the dive just because we made a mistake and dropped something. So in that blue bag, he will collect all of those samples. And then when we go to the top, back up to the ship, we will process all those samples, put them in a preservative, and then I would take them back. This was when I was working at Georgia Tech. So I'd take them back to my lab at Georgia Tech and then process them for genetic analysis. So this is the species that we were looking at. This is in a more shallow picture, in a more shallow area. So they look very orange. This is Tubastria coccinea, orange cup coral. And you can see how there's lots of colonies around. This species does not belong in the Caribbean, not in the Atlantic or the Gulf of Mexico. This species is from the Indian Ocean and the Pacific Ocean. That's where it usually lives. But it was introduced probably through the Panama Canal, and it is spread like wildfire through the Caribbean islands and up into Florida. And it's out in the Gulf of Mexico where those oil platforms were. My job was to investigate how it's spreading genetically. Is it a clone, or how does it spread so fast? One of the locations that I have studied at is Aquarius Reef Base. This is an underwater laboratory. It's located near Key Largo, Florida. And this is where scientists and astronauts live underwater and spend about 10 days doing their research or training. I have done 2 missions in Aquarius Reef Base for other projects, but that's why I know that this orange cup coral was living all over it, because on the outside of this habitat, there's hundreds, if not thousands, of orange cup coral colonies covering the outside of it. This picture down here shows me just kind of measuring this area, and these arrows point to a large, relatively large, they're not big corals, orange cup coral colony. So just in this picture alone, there are several colonies just in this small area. So we would collect samples from this habitat. This is a really cool habitat. If you're interested in that type of thing, you can look up Aquarius Reef Base. If you're looking at it and you see divers and they're wearing white, those are the astronauts. The astronauts train in Aquarius Reef Base to train for weightlessness, but also isolation training, because you can't come directly to the surface from there. This is in about 60 feet of water. The where you live is about 42 feet of water, but you can spend all day diving. You can spend 6 hours at 90 feet. Whereas if we were in a boat, you could only spend about 20 or 30 minutes at 90 feet before you would have to come up. So you get a lot of work done when you're saturated in the habitat like this. So that's Aquarius reef base, and there's lots of this orange cup coral on there. When I go back to the lab, this is what it looks like. When I do my genetic testing, I have different methods to look at the DNA and the RNA of the samples. And then I run them out. This is called a gel. And I look at the patterns and look at these banding patterns. And this is actually a representation of how the DNA is broken up and separated into fragments. And if they have the same DNA sequence, they should have the same fragment patterns. And some of them do and some of them don't. So this is one of the ways we determine if these colonies are clones or if they're totally separate individuals. So that's kind of what it looks like, the end result. And then we would write papers and do big analysis and share this information. So we've done this in Florida, and we've done this in the Gulf of Mexico as well. Another project I worked on, or I'm involved with a huge project in Fiji through Georgia Tech, looking at the effect of seaweeds on corals. When seaweeds overgrow coral reefs, they can kill corals. And we were looking specifically how they kill them and what is killing them, because not all seaweeds do. So this is like a little field experiment. This is underwater. So scientists, marine biologists will can set up experiments underwater, and then they go in and monitor. Just like we would do in a lab, we would set up an experiment, but we're actually doing this in the field. So these are corals that we transplanted onto these little cement discs. And then we attached seaweeds, different types of seaweeds to them. And then my job is to go in after a certain number of days and sample these coral colonies and bring it back into the lab. And this is what it looks like when I'm looking at gene expression. So that means what the coral is doing internally, if they're happy or not happy. There are things called stress proteins. And if a coral is stressed, if you are stressed, you create more of these stress proteins. And this is my way of quantifying, of determining how much of these stress proteins are being produced, and other types of proteins. Things are turned on and things are turned off inside the cells, depending on the environmental conditions that they're in. If they're happy, they're not happy. If they're growing or not growing. So some things are not stress induced. They're just, what the growth season of the individual. So I look at all these little dots. These dots mean something to me. They just look like little dots to you, but red means something, yellow means something, green means something, and their intensity means something. And we translate that into we call this a heat map. That's there's red blocks and blue blocks, and it tells me if things are being upregulated or downregulated. And I look at all those genes and how they're responding, and it tells me a story. It tells me the story about whether the coral is happy or not when it's in contact with the seaweed, or if it doesn't care. Sometimes it doesn't care. It doesn't matter. So when we're in the field, I always show this slide to kids, because sometimes kids say to me, I don't need to learn this stuff. Why do I need to learn this stuff? And I like to put this slide in here because these are 2 graduate students that I brought to Fiji with me, and they're doing a test. There's an expensive piece of equipment in their hands right there, and they're testing for photosynthesis inside the corals. Corals are animals that have plants living inside them, or a plant-like organism lives inside of them. And we can see how much food they're making, how much food that the plants are making inside through photosynthesis. And I put this graph in here because, elementary, middle, high school kids have to make these bar graph things. We still do this as scientists. This is actually a figure from one of my papers showing the effect of a seaweed on corals, and it tells a story, right? These graphs tell a story of what happens. There was no effect here, and there was a big effect here, and we show it in different ways. So even if you think you don't need to know what you're learning, you'll never use it again, you might use it again. We use these elementary level things still as scientists. So the other thing I do is look at the cells themselves. So I look through a microscope. I slice the corals, and I can tell if coral cells are healthy or the algae cells are healthy or not healthy after exposure. So we do a lot of different techniques as scientists to answer questions that we're asking. I'll talk about one more little thing. We look at red algae, and we look for compounds that this algae makes. You know how if you have poison ivy near where you are, I have poison ivy near where I am. If you touch it, it makes your skin break out because they produce this compound that people are allergic to. Well, a lot of plants make compounds so they taste bad. So in this case, fish won't eat them or bacteria can't grow on them. And so our job was to look for these different compounds and see if we can use them for medical purposes, see if we can get them to fight bacterial diseases in humans, or the malaria pathogen, or see if we can kill fungal infections or cancer, different types of viruses using these compounds. And my job in this was not to identify compounds. That's what a chemical ecologist does. They're the chemists that do ecology stuff. My job is to look at the DNA sequence of the red algae so we can identify the species. Sometimes they look so similar that you can't really tell what species you're looking at. So my job was to go in and identify all of the samples that we'd collected, so we have a record of the diversity of what we collected, but also if there was anything interesting, we would know who was making it. So I'm showing you some pictures now of kind of what I do these days. So I'm still involved in science and research, but I try to use that in an educational way. So these are just some pictures of what we do at Discover Science Center and Guy Harvey Ocean Foundation when we work with kids. So we have taken science and made it really fun and easy for kids to understand through a lot of hands-on activities. So we do things like fish anatomy, and they can look at a fish and learn all the different parts. Same thing with sharks. Here's some of our sharks, and we teach the kids anatomy and their adaptations and why some sharks live in some areas and other sharks live in other areas. Those are all the types of things that we're interested in teaching kids, and why it's important to protect the sharks and protect the reefs and take care of our oceans. And I also teach scuba diving, and I primarily teach scuba diving to kids. I teach them how to dive, and I teach them how to do science underwater. So I teach scientific diving. Kids learn how to measure corals underwater, how to collect data, take samples, and all that. So trying to teach the next generation of kids who want to be marine biologists but don't know how to go about doing it. So that's my job, is to try to inspire those kids and give them a pathway to do what they want. And that's me.
Thank you so much. That was amazing.
You're welcome.
I think that you win for coolest office. I'm jealous that you're out there on the reefs all the time.
Yes, here's one. I was just snorkeling with whale sharks a couple weeks ago. So that's a benefit of the job.
Were you able to see the mantas and the whale sharks congregating when you were there?
We saw the whale sharks, but I didn't see any mantas that trip, but I wasn't there for very long. So, but I got some good whale shark time.
It's amazing. So we just have a couple of questions, a lot dealing with the invasives and the relationships that we saw with the corals. So the first one, how different are those genetics between the invasive corals and the non-invasive ones?
Well, so this coral is able to reproduce asexually. So it can produce larvae whenever it wants. It doesn't, so it's a clone. It's genetically identical to that adult. And so it just releases these little tiny larvae. Most corals don't do that. One of the reasons that it can become invasive is because of that characteristic. That's what helps it become invasive. Most corals can't do that. This coral also doesn't have any symbiotic algae that live in it. So it's not restricted to depth. It can pretty much live wherever it wants. It can live in a dark hole if it wants to. That also helps it become invasive. The genetics, so in Florida, the samples that I took, most of them are the same clone all the way down the Keys. But in the Gulf of Mexico, they're very different. There's a huge genetic diversity within the populations there. So I think it's very location specific as well.
Are they sensitive to environmental changes, like how our corals are not adapting so great to climate change? Like, are they more adaptable to that?
So I have a theory. I don't know if it's true. So at a time when most of our corals are dying, are not doing well, this coral is thriving. Like it's taking over. It'll take over whole. The lucky thing is that it prefers artificial substrate. So it's not so much on the natural reef yet, but we do see it on the natural reef. I think the relationship between the symbiotic algae and the coral, that's what is being impacted by the changes, by the environmental conditions. And it's that relationship that is causing the disruptions of that relationship that's causing damage to those corals. Because this coral doesn't have that relationship, it doesn't care. It just keeps growing.
Wow.
It's not dependent on that relationship. That's my theory. I don't know.
Very interesting. So answers to come, right? It's part of science. Kids, you just gotta keep doing that research and collecting that data.
That's right.
So I was really interested in hearing about that relationship and the stressors with the algae. So zooxanthellae are photosynthetic, like we were saying, and they're that symbiotic relationship with the corals.
Mhm.
So with those algae growing on top of them, is there any kind of competition between the algae and the zooxanthellae for those nutrients and things necessary for photosynthesis?
There's probably some kind of shading impact, but we are finding that it's actually a chemical produced by the seaweed, because we can extract the compounds and put the compounds directly on the coral and see the same impact. So it could be, and we do see impact with genes related to the relationship between the corals and the zooxanthellae. So I don't know if it's a competition between the algae and the zooxanthellae, or a response to the chemical produced, because it's not all seaweeds. I can put certain seaweeds directly on it and it has no effect. So in that case, it's not impacting the zooxanthellae at all, at least in any way that we can quantify with the genes that we're looking at. We're looking at thousands of genes, so we would think we would see something. But I think it has to do more with the specific chemistry of whatever that compound is and how it affects. Some of these compounds, their job is to kill things. So the red algae doesn't get overgrown, right? So there's competition there. So they create these, even that orange cup coral, it secretes a compound that kills everything around it, so the babies can land and grow. So it's in habitats where space is very limited, you try to prevent things from growing near you and on you. And that's how they do it, through chemistry.
So interesting. And then we have just two more questions. Mm-hmm. I know that a lot of students are going to be interested in the scientific diving and diving as a career. We have a ton of kids that dive for fun with their family. So how do you become a scientific diver?
So this is actually a tricky question, because there isn't a true scientific diver certification that goes to everyone. You become a scientific diver by working for an organization that has a scientific diving program. You get trained by them. You have to meet some certain requirements. However, you can take courses and classes and do pool sessions to learn the techniques, to learn the methods. But it doesn't, it lets you learn how to do it, but it doesn't certify you as one to go work wherever you want, if that makes sense.
Yeah.
So number one, you have to dive, because all we're doing is teaching neutral buoyancy and doing tasks while you're neutral. Like that's all it is task-oriented diving. So if you've got diving down, then you can just pick up something and start doing it. You just need to learn those methods. But all that diving practice is needed to become a good scientific diver, because you can't think about your diving anymore.
Mhm.
That's second nature. You're automatically looking at your air. You're automatically looking at your depth without even thinking about it. You're neutral. And then you can focus on your tasks. So it's just practice, practice, practice. You have to find someone who knows how to teach that and is willing to teach that. And then you need to work for an organization who certifies you and puts you out on the research boats. It's kind of complicated.
Yeah, so it sounds like get a lot of those dive hours done and just be an expert by the time you're ready to finish university, and then kind of find your way into an organization that requires that.
Absolutely. Like a lot of grad schools will have a scientific diving program, and it's part of in the United States, at least, it's the AAUS, American Academy of Underwater Scientists. And that's who organizes all of these dive programs at different universities and field stations and research institutions. And then you become one of their divers, but only for that institution.
Great. Well, thank you so much. We will be sure to link all of the places that Dr. Shearer is associated with in her activity and on her landing page for this program, so that you guys can check out all the cool stuff that she's doing and follow along and get more information. But if you would like to participate in a research project, maybe do your own investigation on corals, we do have our own research experience program. We're currently accepting application or enrollment for fall, and we will be releasing spring and summer soon. So definitely get on board with that if you would like to do your own research. As you see, science takes you all over the place. I mean, you can be on coral reefs all day, every day, taking samples and doing the research that way. If you would like to have any more questions answered, you can email me at jen@headwaterscienceinstitute.org. So thank you again so much for spending time with us today. We're excited to get your activity out so kids can participate in doing their own kind of investigation looking at coral reefs and invasive species.
Very good. Thanks for having me.
Want to learn more about our Lunch with the Scientist program? Make sure to like and subscribe and visit us online at www.headwaterscienceinstitute.org for a full list of our video library and curriculum.